Talked about old school genetic engineering, selective breeding. Now we're going to talk about new school. Now, new school deals with the actual manipulating, the changing of the DNA. And so, first off, before we get too far into this, we have to first talk about what exactly is genetic engineering. So, genetic engineering is simply the changing of the DNA code, okay, by making those changes. To do that, we're going to have to be able to go in and cut stuff and move stuff around. To cut DNA, we use something called restriction enzymes. Okay? A restriction enzyme is an enzyme that cuts DNA at a very specific sequence of nucleotides. Here we have an example of a restriction enzyme that cuts DNA at the sequence of CTTAAG. Okay? So anytime it sees that CTTAA, it will come and it will cut it right at that spot. So when that happens, because it doesn't say, oh, I'm looking for this specific gene, it'll cut the DNA anywhere along that sequence. Now, so it takes it and it cuts it. Only if it sees that sequence. Now, we have all these different cuts, right, these different size lengths of DNA. This is where gel electrophoresis comes in. Now, the overview here is what happens. All right, we cut it, we place it, and it moves along this gel-like substance. So what happens is, the first step is that we take restriction enzyme to cut the DNA into smaller parts. Okay, we then take this DNA, these fragments, and put them into these wells, all right, along the gel. Once that happens, the next step is to apply a voltage. All right, we apply electricity. Now, DNA actually is polarized, and so when we add that current, the DNA will move towards the positive side of the gel. So it moves from where it started away from it. Now, the smaller pieces, remember back from your physical science, smaller pieces, when you apply the same force to it, will move faster and therefore farther along the gel than the thicker pieces. Right? I apply the same force to a tennis ball and a bowling ball. Which one's going to move farther in the same amount of time? Well, the tennis ball is, because it has less mass. So, because of that, when you take a look at the gel electrophoresis, you can see that the shorter fragments are farther from the beginning, and the longer fragments, ones with more mass, are up here closer to the beginning. From this, you can see a bunch of strands, or these bands here. Right? We then use and compare these bands to other samples' DNA to gather information from them, to see if they're the same DNA, etc. Now, researchers then can use, like I said, these similarities and differences in the genomes of different kinds of organisms to see how closely related they are, etc. Now, well, also we use gel electrophoresis to help find the sequence of DNA. So now, in brief, what happens is this: you take DNA, you cut it, and you attach dyes to the last end, and these nucleotides with dyes then get organized by length, and then you can then find the sequence by reading the different colors of DNA, because each nucleotide will have a different color. So then you can read the sequence from smallest to longest, and you can get the correct order. So that's how they're finding out the A's, C's, T's, and G's and where they all belong. Now, when we take these enzymes, it's possible then to take DNA, once we have the sequence, and combine that DNA with other DNA. This combining of different sources of DNA is called recombinant DNA. And one of the processes I want to talk about real quickly is something called polymerase chain reaction, or PCR. And all this is taking a strand of DNA and replicating it super fast, that you have a bunch of stuff to work with. And here it simply takes, as you can see in the diagram here, you go from 1 to 2, and it increases exponentially the number of strands of DNA that you need. Now, besides that, I want to talk about something called cell transformation. Okay, and transformation is when a cell takes in DNA from outside the cell, and then it takes that DNA and makes it its own. One of the things I want to talk about also is something called a plasmid. A plasmid is simply a circular DNA molecule. We're going to take a look at a diagram here and explain exactly what takes place for this to happen. So here we have a human cell, okay? And there's a human gene for a growth hormone, all right? Say insulin, okay? So for the longest time, we were using, for those that are diabetic, now a person that's diabetic has a hard time accessing glucose. And so a diabetic was often given pig insulin, and because pigs are very similar to humans in many ways. Well, the problem is that if you inject something that doesn't belong in your body, if you remember back to our blood unit, or talking about blood, the body reacts to that negatively. So what has happened is the scientists said, well, can we find a way to grow human insulin? And so what they did is a process similar to this, where they took a human gene for creating insulin, all right? They attached it to a plasmid. Here's this circular piece of DNA. They cut the plasmid open. They stuck the DNA of the gene into the plasmid and then took that plasmid and injected it into a bacteria cell, where then the cell bacteria takes up that DNA and then masks it or makes it its own. Now all of a sudden, a bacteria cell that was just a bacteria cell now is a bacteria cell that will then start to produce that human insulin. And then the people can then harvest it, and you have a cheaper version of insulin that isn't going to have the reaction problems like pig insulin. Well, transformation is considered successful when that recombinant DNA is completely integrated into one of the chromosomes of that new cell. Now, transgenic. What does transgenic mean? Transgenic means an organism that contains genes from other species. And now if we take a look at a lot of the foods that we have, we're getting into the point where we're taking genes from one organism and putting it into genes or into the chromosomes of another, so that the benefits of one organism are now seen in the other. We're taking a look at in the future of having plants that are resistant to certain bugs, resistant to the different types of weather like cold, etc. Last thing I want to talk about real quick here is cloning. Now, a clone is simply when a member of a population of a genetically identical cells produced from a single cell, right? So a clone is a member of a population of genetically identical cells produced from a single cell, right? Way back in 1997, there was something called Dolly. And what happened here was they cloned this sheep. Now, this is the process that takes place. The first thing that takes place is you have a cell taken from a sheep, all right? The donor cell, the organism that you want to clone. Then what happens is you take another cell, an egg cell from another sheep here, adult female here, and we need to remove the nucleus, because if we added the donor nucleus and had the egg nucleus, we'd have too much genetic information, we'd have problems. So what happens is we get rid of the egg's nucleus, genetic material, and then we just add the donor material. So now we have the egg cell with the donor material in it. Next, what happens is that fused cell is then allowed to divide, and from there it creates an embryo. Now that embryo is then placed within a foster mother where it's allowed to grow, and then finally you have your cloned animal looking just like the original donor. So from this process right here, remember, you have the donor cell, can be a skin cell, whatever, egg cell from another animal. We have to get rid of the egg's genetic material, that donor material is fused in, it's allowed to grow in a foster mother, and then a cloned animal is finally done. So there we go, Genetic Engineering 101, the very, very basics. Hope you understand. Questions? Good luck. And stop.